Hi, class. It's Dr. Lindner. Welcome. In this lecture, we're going to be talking about the neural system and neurophysiology. It's a topic that a lot of students really don't like too much. I'll try and keep it as direct and simple as possible. So let's just do a quick overview of the neural system. So here up at top, we see we have the central neural system. And remember, the central neural system is going to be the brain. So let me go back here. Sorry about that. Let me get my pen on. It's going to be the brain and the spinal cord. Okay. So if we have the brain and the spinal cord, pretty simple. And some basic concepts is that the brain can send information going down the spinal cord and out, and we're going to call that motor information. And then there's information that can come into the neural system, into the nerves, into the spinal cord, go up to the brain, and land there, and we call that sensory information. All right, so we have input and output, right? We have incoming information, we have some processing, we have outgoing information. The sensory information, we know that it ascends because it's going up, and we call that afferent information. The information going down, it descends, so we call it descending pathways, descending information, and it's going out, so we call it efferent information. So we have sensory information coming in, it ascends, it goes up, and it's called afferent. Motor information goes down, it descends, and it's called efferent. Just some basics. So we have sensory and motor divisions, right? We have sensory division and we have a motor division. From the sensory division, we have information coming into the brain and the spinal cord, the CNS, from your somatic senses, from all of your body parts, your joints and your muscles and your tendons, right? And your special senses, what you smell and what you can touch and what you feel and what you see and what you hear and all that information, all of your proprioceptors, they come into the central neural system and you have processing. And now there has to be motor output that goes to your somatic neural system. Your somatic neural system is going to your skeletal muscles. And the motor division could be the ANS, the autonomic neural system, primarily sympathetic and parasympathetics. For the most part, we think of the sympathetics as the accelerator, and we think of the parasympathetics as the brake. These go to sympathetic and parasympathetic, go to all of your internal organs, things that have smooth muscle, your heart, your cardiac muscle, your glands, all of your gastrointestinal tract, all of your internal organs have sympathetic and parasympathetic. When you're being chased by a tiger, the sympathetic neural system, we call it fight or flight, where your heart rate goes up and your breathing increases and your pupils dilate and your digestion shuts down. And when your parasympathetics are stimulated, your blood pressure goes down and your breathing slows down. This is more like feed and breed, more of your relaxed state. But your digestion is activated and speeds up with parasympathetics. Okay, it's a little bit of a breakdown. We know that the sympathetic is called the thoracolumbar output because it goes from T1 to L2, and the parasympathetics are referred to as the craniosacral output because it's cranial nerves 3, 7, 9, and 10, and sacral segments S2, 3, and 4. And just to review the different parts of a neuron, because we talk about neurophysiology, we have action potentials that are going to be taking place traveling through a neuron. So the different parts of a neuron, we have the dendrites, we have a cell body with a nucleus, we have an axon, and at the very end, we have an axon terminal with these synaptic end bulbs. and the functions of each, right? The electrical conduction is going to be traveling in this direction, like here. So the dendrites receive the stimulation through activation of either chemically or mechanically gated ion channels. So in this lecture, we're going to be talking about chemically and mechanically gated ion channels. So, gated, when we have a cell, the nucleus, and we know it's a phospholipid bilayer, we have channels like doorways for things to enter or exit. And there's going to be gates, like, think of it as two doors. There'll be one door or one gate here and one gate here. So things can enter, like sodium can enter through this inactivation gate, come in, but it's not in the cell yet. It just leaves the extracellular. Now it's kind of like hanging out in the cell membrane. Then the second gate or the activation gate opens and boom, it rushes in. So these are the gates and these are the channels. And again, there's typically two gates located in the channels. Some textbooks just show one gate for simplicity, but just to give you an idea. You're going to hear of terms called excitatory postsynaptic potential and inhibitory postsynaptic potential. Okay, so the cell body receives the stimulus and produces excitatory postsynaptic potentials or inhibitory postsynaptic potentials. And you will also be hearing words like depolarization and repolarization. Depolarization, repolarization, and hyperpolarization. So you're going to be hearing these terms that I'll go over and show you what they are. So then the information is still moving down through the axon hillock, which is over here. There's a trigger zone where we see depolarization can take place if it reaches threshold. That's another word that I'll go over with you. So some key words: depolarization, repolarization, hyperpolarization, threshold, action potential, resting membrane potential. These are all terms that you have to have a strong understanding of, and I'll go over in this video. So main parts of a neuron, and we'll go over these throughout the video. So let's talk about the electrical signals in neurons. Now, neurons are electrically excitable due to the voltage difference across their membrane. And there's communication with two different types of electrical signals. One we call an action potential. Sometimes you'll see it abbreviated as AP for action potential. And action potentials can travel very long distances, whereas graded potentials, these don't. These are just creating a local membrane change only. And in living cells, a flow of ions occur through ion channels, and these ion channels are in the cell membrane. Is it a sodium channel? Is it a potassium channel? Is it a calcium channel? So this is I want you to get comfortable with some of these numbers. Let's see if I can just change the color here so not everything shows up black. Let's use this blue color here. So few numbers that I want you to keep in mind. Let's keep in mind this -70. Let's keep in mind this purple line right here. The purple line is right at around -55. That's what we call threshold. So we have -70. -70 is called resting membrane potential. Nothing's really happening here. See this line? -70 is a resting membrane potential. But you see how right here it started at -70, then it kind of went up a little bit to, like, I don't know, what is that, -67, 68, then it comes back down to resting membrane potential, then it comes back up a little bit to maybe -68, then it comes back down. So these little local changes, those are called graded potential. Okay, that's a graded potential. An action potential only happens when this minus 55 is hit. Okay, when that minus 55 number is hit, so this is still like it's changing. It's going from resting membrane potential. It's going to like minus 65, minus 60. Something happens at minus 55, and I'll go over what that is shortly. But something happens at minus 55 where a threshold is met. And then look what happens. Boom. This crazy change takes place and it goes up to positive 30. And then something happens here at positive 30, and I'll lay down some framework. Is that, remember the cell, the phospholipid bilayer, and we have these channels with these gates. So something has to be rushing in to activate, and then there has to be another channel where certain things exit. So something's happening at this minus 55, something's rushing in that creates this change, and then maybe these channels or these gates close, boom, and it moves back down to the opposite direction because maybe something is leaving the cell, and it creates this change. Depolarization, I'll put a D here, is when it's moving away from the resting membrane potential in a more positive direction. And then repolarization is when it's descending, right? It's coming back down. It's moving in a more negative direction towards resting membrane potential. That's repolarization. But before I said there's depolarization, repolarization, and hyperpolarization. Hyperpolarization is when, see how you have minus 70 right here, which is resting membrane potential? Hyperpolarization is as it's coming down, sometimes you'll get a dip in a more negative direction for a little bit, and then it comes back up to resting membrane potential. So when it dips in a more negative direction below resting membrane potential, that right there is called hyperpolarization. So you have, let's review, we have some graded potentials, which are just a local change. It's graded until we hit threshold, which is minus 55. And minus 55 happens because there's a lot of sodium out here. There's lots of sodium extracellularly. And some of the sodium goes through this first little gate. And when that first gate is open and some of it sodium leaches into that cell membrane, it starts to move in a more positive direction, right? Because it's going from minus 70 and then it's going to minus maybe 68, 67, 60. And then when it hits 55, this other gate opens and all the sodium rushes into the cell. All of this sodium moves in, and boom, we get this spike till we hit positive 30. Then both these gates close, and potassium that's in here starts to exit. And then that creates this, boom, this repolarization. And it moves in a more negative direction to resting membrane potential. But the potassium gates close a little bit slower. So as the potassium still leaks out, we get this repolarization where maybe it dips to minus 80 or minus 90. And then the sodium potassium pump kicks in and starts to rebalance things to bring us back to resting membrane potential. I'm going to go over that a few more times, but that's a key. And as we're moving up in this direction, this creates an excitatory postsynaptic potential, and on the way down, that's an inhibitory postsynaptic potential. Okay, this is more sodium related, and this is more potassium related. So now we have these different types of channels. We have leakage channels, and then we have gated channels. Leakage channels, these are non-gated. They're always open. It's kind of like those old-fashioned hoses that have those holes where people used to, put them around their gardens, and they turn on the spigot and the water comes out of the hose and it's permeable, and that's like it's non-gated. And it's important to know that nerve cells have more potassium channels than they have more sodium leakage channels. And as a result of this, it creates a membrane permeability that's higher to potassium. And as a result of that, it creates this difference, or this RMP, this resting membrane potential of minus 70 millivolts in nerve tissue. On the gated channels open and close, and they open and close in response to different types of stimulus. Now we have voltage gated channels, we have ligand gated channels, and we have mechanically gated channels. Voltage channels, these are going to open and close in response to a change in the actual voltage. When you hear ligand gated, this is going to refer to some sort of chemical stimulus, like a hormone or a neurotransmitter or an ion. And mechanically gated, this is based on pressure. This is a vibration, something mechanically, some sort of mechanical input to the body. So when we look here at the top one, we can see this is going to be voltage gated. We could see that at minus 70 millivolts, this gate is closed. We see extracellular and cytosol, which is intracellular. And what happens is that from minus 70, now there's a change in voltage to minus 50, this channel opens. And what's happening, it looks like this is a potassium channel, so it's allowing potassium to go from inside the cell to outside the cell, right? Simply put, we have more sodium outside the cell, we have more potassium inside. So things tend to move down a concentration gradient where sodium wants to move in because it's going from higher to lesser, and potassium will want to go out because it's going from, again, higher to lesser. Okay, but then you have the sodium potassium pump, which is moving it now against its concentration gradient, and that's what requires ATP, requires energy in order to do that, to restore it back to its RMP, its resting membrane potential. Now on the bottom, notice we have chemical, because this is ligand gated. We have something here like a neurotransmitter, these red little pyramids here, that's acetylcholine. When acetylcholine binds to this channel, open says me, right? It binds, and now when it opens up, we have things that can move in both directions. So it looks like we have sodium moving in and potassium moving out, right? It looks like the green ones here are sodium, and then the pyramids there, those are potassium. So we have voltage gated and we have ligand gated. We're going to see these come to work shortly. And this happens quite a bit, right? When we talk about muscles and we talk about the neuromuscular junction, we're going to see this take place. We're going to see acetylcholine bind to the sodium channel. Boom, sodium rushes in and initiates an action potential to make muscles contract. So that's kind of an important thing. Now we have mechanically gated channels, and these respond to mechanical vibration or pressure. This is really important for us. So we have some sort of mechanical stimulus that will open up the channel, and you have, again, you have things like calcium moving in or sodium moving in. And again, this is really quite important when we talk about muscle contractions, which involve pressure. And when you walk, think about the pressure on the soles of your feet, which help initiate the gait cycle. So that's an important concept. In terms of RMP, resting membrane potential. We have negative ions along the inside of the cell membrane and a more positive ions along the outside of the cell membrane. Okay, so we have the cell membrane like this. We have negative ions along the inside, kind of like this. And we have more positives, more here. And remember, positives and negatives attract, so what's outside goes in and what's inside goes out, right? Things are always moving in these opposite directions between sodium and potassium. But that difference, the potential difference between both, is that minus 70 millivolts. At rest, at resting membrane potential, the concentration of ions is different of what we have outside and inside. So in the extracellular fluid, everything out here, we're going to have lots of sodium. We're going to have lots of chloride out here. So we have sodium, we have chloride on the outside, and on the inside, we have more of potassiums. And we have more protein, more amino acids on the inside. And the membrane permeability is different for sodium and potassium. It's about 50 to 100 times greater permeability for potassium, so that inward flow of sodium can't really keep up with the outward flow of potassium, right? It's kind of like water, like a hole in a rowboat, and the water keeps coming in. And to avoid the boat from sinking, you have someone that's taking the water and they keep pushing it out of the boat into the ocean, and then water keeps leaking in, and you have someone that keeps taking the water out of the boat, pouring it back into the ocean. This is that sodium potassium pump that's trying to remove sodium as fast as it comes in, right? Sodium is rushing in, potassium is rushing out, all right? The inward flow of sodium just can't keep up with that outward flow of potassium because there's more potassium leakage channels. So the resting membrane potential, that the membrane of a non-conducting neuron is positive on the outside and it's negative on the inside, right? Remember, we drew this, was more positive here and a little bit more negative here. So the resting membrane potential is determined by the unequal distribution of ions across the plasma membrane and the selective permeability of neuron membrane to sodium and potassium. Most anions cannot leave the cell, right? You should be familiar with what anions and cations. Anions are the negative and the cations are the positive. Just remember, cations, the T looks like the positive sign. And of course, the sodium potassium pump. So those are the three things that are helping to maintain that RMP, the resting membrane potential. So here we have the extracellular fluid. Here is the cytosol, intracellular fluid. This is a phospholipid membrane. These are the phosphates on the outside. Again, you have phosphates on the inside, and you have the lipid tails here. Out here, this is hydrophilic, and in the middle is hydrophobic. And when they put in a meter to look at what is the resting membrane potential here, you get a minus 70 millivolts. Again, more positive on the outside, a little bit negative in the inside. Positives and negatives attract. So what's here moves this way, what's here moves this way. We have sodium rushing in, we have potassium moving out, but then to restore this, we have that sodium potassium pump that's designed to keep things at RMP, the resting membrane potential. So we have potassium leakage channels, sodium leakage channels, right? So look what's happening. From here, we have potassium leaking out, and from here, we have sodium leaching in. Okay, and then what do we have here to try and restore things back to balance, to resting membrane potential? As the sodium rushed in and the potassium rushed out, now we're trying to restore things. but it does require ATP to push 3 sodiums out to bring 2 potassiums in. It's 3 out, 2 in, 3 out, 2 in. That's the sodium potassium pump. Now you heard me talk about graded potentials. Now here's our little scale. Here's -70. That's your resting membrane potential. We see it here and here. And when it starts to move in a more positive direction, right, where certainly we're moving in a more depolarizing, but it never hits threshold. So that's a graded potential. When it moves below -70, that's hyperpolarization. Okay, these are graded potentials. They're just local changes. But if it doesn't hit -55, remember -55 is threshold. If it doesn't hit threshold, then it doesn't depolarize all the way up to +30. So these are just small deviations in a resting membrane potential. Okay, so we talked about the graded potential occurs in response to the opening of mechanically gated or ligand-gated channels. This is nothing new, whether it's applied pressure or, right, the mechanically gated or vibration and pressure, that can open up the gate, or we have ligand, which is the chemicals. So whether it be the neurotransmitter acetylcholine or whether it be glycine, these have the ability of hitting the channel and then opening up the door. Okay. Now, some are excitatory and some are inhibitory. When we move towards -55 or threshold, that's going to be excitatory, and acetylcholine is an excitatory neurotransmitter. So when you see depolarizing, that's when we're going from, let's say here was -70, then we had -55, 0, and +30. So we go from here, resting membrane potential, it moves up slow. Once it hits that -55, boom, we get that rapid spike. That's because there was an excitatory neurotransmitter. But certain neurotransmitters are inhibitory, like glycine. That's inhibitory. So what that's going to do is that's going to create hyperpolarization. So that's going to create it to go boom, it's going to move it below resting membrane potential. -70 is resting membrane potential. This dip right here, that's hyperpolarization. Now it looks like it's probably -90 or somewhere. So that hyperpolarization, it moved in a more negative direction away from resting membrane potential. Those are inhibitory neurotransmitters. So an action potential is a sequence of rapidly occurring events that decrease and eventually reverse the membrane potential and eventually restore it. So that that's what we call depolarization, and eventually restore it to a resting state. That's repolarization. Okay, so again, if this is our -70 is resting membrane potential, -55, that's your threshold, and we have 0 and then +30. So here's resting membrane potential. Now we have a graded potential until we hit -55. We depolarize to +30. Then we're moving back to resting membrane potential. That's repolarization. And if it dips below -70, that's hyperpolarization. So we have a graded, we have depolarization on the way up, repolarization on the way down, and hyperpolarization if it dips below that resting membrane potential of -70, right? So here is hyperpolarization. Here's a graded potential. Hasn't hit -55 yet. So hyperpolarization is when the membrane becomes more negative in relationship let's be clear about this, in relationship to the resting membrane potential. Okay, that's the key. Hyperpolarization of the membrane becomes more negative in relationship to the resting membrane potential. Here, the resting membrane potential minus 70, when it becomes more negative, that's hyperpole. Not going from positive 30 in a more negative direction to RMP, that's not hyperpolarization, that's repolarization. When we go below RMP, that's hyperpolarization. Depolarization is when the membrane becomes more positive. So here, from minus 70 to minus 50, that's in a more positive direction. From minus 55 to 0, that's in a more positive direction. From 0 to positive 30, that's in a more positive direction. So hopefully that makes a little bit more sense now to you. So here, again, we've got our local or graded potentials. Now, sodium, one of the sodium gates here starts to open. So it moves in a more positive direction. Once we hit positive 55, not positive 55, minus 55, I should say, that's threshold. Both of those gates open and sodium can sneak through both gates, the inactivation gate and the activation gate. When one of these gates are open, sodium can rush in through the inactivation gate, that first door. Now it's hanging out in the cell membrane. So maybe it's going from minus 70, maybe it's minus 60. And when enough sneaks in through that first door, it will hit minus 55. And that's when this one opens up. Boom. Now lots of sodium runs in. And that's what brings it up to this quick positive 30. Then the sodium gates shut. Boom, boom. Potassium opens, rushes out. We get this dip. Okay. All right, let's run through this again. So let's look at the chart. We see a minus 70. That's our resting membrane potential. We see this local change here, but now it hits threshold, minus 55, and then boom, it depolarizes up to positive 30. Then something happens, and we repolarize, and then, ooh, looks like it dips below minus 70. We have hyperpolarization. Notice we have these different colors. Each of these is a different color. And we have the color coding of what's happening off to the right. So there's a series of rapidly occurring events that change and then restore the membrane potential back to its RMP, its resting membrane potential. So ion channels are open, sodium rushes in, that's depolarization. When potassium rushes out, that's repolarization. There's this principle called the all or none principle that says with stimulation, this either happens one specific way or it doesn't happen at all. It's the all or none principle. So in this phase right here, in the purple phase, resting membrane potential, voltage gated sodium channels are in the resting state, and voltage gated potassium channels are closed. So this is what's happening at RMP, at resting membrane potential, right? You got nothing really happening at the sodium channels. They're in the resting state, nothing really happening at the potassium channel. But what's happening in the blue? Some stimulus causes the depolarization to threshold. So some sodium was rushing in through one of the sodium gates, okay? So let's look here. Right here, here is your sodium. Here is your nerve cell. Here is the sodium channel, green, and then here's a potassium channel. So we're going to have like one gate here, and then here's another gate. So if you have some sodium, remember RMP is minus 70. But as some of this sodium goes through that first gate and it's hanging out here in that cell membrane, that minus 70 maybe goes to minus 60. Okay, to minus 57, to minus 66. And once it hits minus 55, which is minus, I should call this minus 55, that's threshold. And now, boom, this gate opens right here. And now you got all the sodium rushing in, and that's what brings us up from minus 70. We get this little bump here till we hit that, as the sodium moves in here, but not into the cell, just into like that membrane, it's making that local change. But once enough pumps in to get us to that minus 55 or threshold, boom, this gate opens, sodium rushes in, we hit minus 70. And now this closes and this closes, and potassium channel opens. And as potassium rushes out, we start to get this repolarization to minus 70 to resting membrane. But the potassium channel shuts in a more delayed response. It's not as quick as the sodium because of the delay. Sometimes you get that hyperpolarization. Okay, so chemical or mechanical stimulus causes a graded potential right here to reach that minus 55 threshold. And the voltage gated sodium channels open, and now sodium rushes in. And when sodium rushes in, boom, we get that intense spike up to positive 30. Okay. Let's go back here. So now on the repolarization, let's see if I can get this back. Okay, so what this is saying here is that when 55 is hit, what happens is the potassium channel opens. So right here, right there, now the potassium channel opens. And that's going to cause this, see if we can draw it here. It's going to cause this to spike down. So sodium shuts and potassium opens. And when that happens, that's going to work towards repolarization to restore us back to minus 70. But the potassium channel, there's a delay in the closing. So we get this hyperpolarization. This happens because of the delay in the potassium channel closing. Okay. So to review, sodium channel opens, boom, we get the spike for depolarization, sodium channel shut, potassium channel opens, potassium rushes out of the cell, and because there's a delay, we get that hyperpolarization. Now you can get another action potential that creates another depolarization, but that can only happen when the sodium channels are fully closed. So the period of time during which a neuron cannot generate an action potential, it's called an absolute refractory period. Okay, the inactivation sodium channels must return to the resting state before they can ever reopen. Even a very, very strong stimulus can't initiate another action potential. So in an absolute refractory period, you absolutely cannot have another action potential. So that means when you're in this state, you can't have another action potential. But in a relative refractory period, a super threshold stimulus will be able to start another action potential. All right, the potassium channels are still open, but the sodium channels have already closed. That's really the trick. When can another action potential take place? Only when those sodium channels have closed. In the absolute refractory period, you absolutely cannot have another action potential. So the action potential summarized, the RMP was minus 70. Depolarization is when you're going from minus 70 to positive 30. That's when you depolarize. And that's because, remember, that minus 55 was hit, which is threshold. Repolarization is when you're going from positive 30 back to the minus 70. And then what is it called if you go from minus 70 to, let's say, minus 90? That's called hyperpolarization. Okay. Okay, let's take a break here. When we come back, we'll talk about the propagation of action potentials.